time to talk about mutations. So, what happens when genetic information like your DNA or your mRNA is not copied correctly? So, I don't know, this was some show that I watched, Teenage Mutant Ninja Turtles, and I've heard they've made a comeback. Were famous because they stepped in some water and they mutated, and these turtles can talk. So what causes mutations? So we're going to take a look. We have an image here. There's different things that we know cause mutations. Radiation's one of them. So, too much UV radiation and X-rays can induce mutations. Chemicals, we're aware that like cigarette smoke, nitrates, even too much barbecuing, some products in our medicines, they can actually cause mutations. And there are infectious agents. So some bacteria and viruses can actually cause mutations. Now, when we talk about chemicals, I usually have students ask me about different chemicals, and we have to kind of weigh the pros and cons of all of these things. For example, you're going to need X-rays at some point. So you just don't need them every day. So they are cautious on how often they do this. So just kind of keep those things in mind. Also knowing that mutations can happen spontaneously, which means just they can happen at any time. So there are two types of cells in your body. So this is a review. Cell types. Somatic cells are your body cells, like your skin, your hair, your heart. Now, if a mutation happens in those cells, it might just be a little effect or very little impact because it's only going to impact that cell and future cells of that cell. So, for example, when it goes through mitosis, it's going to pass that same mistake on to those future cells. So it's going to tend to be localized. Now, those mutations cannot be passed on to your offspring because your skin cell, where there might be a mutation, is not going to be passed on to your future offspring. However, when I say that they can have little impact, it just depends. A brain tumor is oftentimes a mutation in just your brain tissue. But if that brain tumor actually that mutation, those cells mutate and there's a tumor accumulates, they could have a large impact and could affect your quality of life or the length of your life. So we want to make sure we just kind of understand that typically, we all probably have several mutations in our body, but it's only if they actually impact one of, an organ that's vital, so we use the term vital, saying like it's something necessary for life. Now, germline mutations occur in sex cells. So that would be your sperm or your egg. So those mutations can be really significant because if there's a mutation when those cells are being formed, then that mutation will get passed on to the future offspring. So we're talking about mutations and it's kind of sounding negative, but mutations really lead to variety and diversity. So not all mutations are bad, but we're going to take a further step here in looking at types of gene mutations. So there's two types to add to your notes. The first type we're going to talk about are substitution or point mutations. So they happen at one particular base, and it's oftentimes something substituting in for something else. So it's like you take one, you give one. Okay, the second type will be frame shift mutation. So I want to leave our room in our notes to talk about each of those. So let's start with point mutation. So for example, if there's no mutation at the DNA level, it should be TTC. The mRNA, that should be AAG, that should code for the amino acid lysine. Now there's three types of point mutations. One thing that could happen is that instead of this C, there could be a mutation and the nucleotide T would be here. And that would change the mRNA codon to be AAA, which is still lysine. In that case, we'd say the mutation is silent because there really is no change. It's the same amino acid. Not so bad, right? Well, moving on, if there is a nonsense mutation, in this case, maybe the A is now mutated and is there instead of the T, This is going to create the mRNA code UAG, which says stop. That's a problem because the amino acid that should be there is not there. Other possibilities are missense mutations, where you could have the possibility where you have TCC or AGG, which would give you arginine, still the wrong amino acid. And we talk about this non-conservative. This is also going to be an amino acid. Now, the difference between these is that at least this amino acid is basic. This one is apolar. So what basically proteins are going to fold and they're going to be attracted. So this one, even though it's a missense, may not have as big of an impact, but this one's not as good because it's not going to bond properly. And we talked about how proteins have to be folded, and that's what's important here. If proteins unfold or they don't fold properly, we say they denature, and we know they won't work. So the second type of mutation that we know happen in gene mutations are frame shift. So this is really just a practical example. Read the sentence. The fat cat ate the rat. That's what the sentence should normally say. Now, if we delete this A and we try to read the sentence, regrouping the letters in three, the fatiz ata tata herat doesn't make any sense. So oftentimes frame shift mutations shift the entire reading frame and cause anything after where the deletion was to create just something that doesn't make sense. The other possibility, instead of a deletion, could be an insertion. So if we insert another A here, regrouping the letters, the fataka ata ete arata doesn't sound like the fat cat ate the rat, does it? So now these insertion mutations also can mess up the entire reading frame. Here's what it looks like if we go back to using our mRNA codons. And this is the normal protein, but this is an example of a frame shift mutation where there was a deletion right here. You can see the arrow pointing out that the U's removed, and then it just shifted the entire reading frame, and it stopped too soon. So if we've just finished talking about gene mutations, that's just impacting one gene. So it could be the gene for hair color. And okay, maybe there's a new gene for hair color, or there is no coloring of hair. Chromosomal mutations, not just one gene, but several genes. So you can look at these pictures and you can see entire segments of chromosomes. So it could be several genes. It could be hundreds of genes are now being impacted. So these terms really are, the self-explanatory. Deletion would be entire segments of chromosomes being deleted. Duplication would be entire segments being duplicated. And inversion would be flip-flopped. Insertion would be taking one from one chromosome and adding it to the chromosome of another. Translocation would be kind of flip-flop or switcheroo of two different types of chromosomes. What you'll notice is these top three only involve like the same chromosome. Insertions and translocations start to impact at least two different chromosomes. I'm hoping that you're wondering, well, why would adding more information, or why does it matter where it is? Those are things that scientists want to really understand too. Someone with a duplication just has extra information, but that seems to cause significant problems. So when we start to look at genetic disorders, they're oftentimes caused by either a gene mutation or they could be a result of chromosomal mutations. And ultimately, any of these mutations is going to have some impact. We can say it's either an advantage or a disadvantage, and it would be looking at individual case-by-case mutations. I hope that information is helpful. There is a video to watch and take notes as you wrap up this activity.